Hello everyone. Welcome to Lesson 3 of Unit 1. Today we're going to be looking beyond Mendel, I suppose, and understanding some forms of inheritance that he was not aware of. One of the most important skills we can bring into this lesson, as you will see, is the ability to do Punnett Squares. So if you haven't yet covered Lesson 2 and practiced Punnett Squares, you should do that before you attempt any of the Punnett Squares involved with this lesson. Be warned. Most people don't know this, but the first organism that Mendel studied wasn't actually the pea. It was a weed called Hericium. Hawkweed is what it's commonly called. And he quickly abandoned it and switched to peas, which was lucky for him because he was able to find a pattern of inheritance that was quite easy to understand. If he had crossed any parents, hawkweed parents, he would have discovered something very unusual. Here we see the results of a cross between two plants. Here's one parent, and here's the other. Okay, and then these are all the offspring. Okay, within here you can see every possible variation of yellow all the way to mostly red. Okay, so you can imagine poor Gregor being utterly perplexed by this result, if he had done it. So clearly there's something going on here which cannot be explained by a dominant versus recessive allele. And indeed, that's the case. There are other ways that alleles can work with each other than just one being dominant and one being recessive. And that's what we're going to be looking at today. The first form of inheritance we need to know is the one that we've already talked about. This is Mendel's form of inheritance, and it's called complete dominance. Something that is complete is total. And what it's talking about is the fact that the dominance completely dominates the other allele. Okay? So if we use the analogy of an A allele that's yellow and a B allele that's blue, if you put A and B together, the offspring are yellow. Okay? They are completely one trait. Yellow in this case, we're going to say is dominant over blue. Okay? So that's complete dominance. All of the different traits that Mendel looked at showed complete dominance, and that's how he was able to figure out the laws of inheritance. Good thing for him. The gene that we looked at last time was flower color. Purple, in this case, the purple allele is completely dominant over the white allele. So if you have a purple and a white, you are purple. There's no whiteness that shows because it's completely, totally covered up. Okay? Complete dominance. The next form of inheritance is called incomplete. If you add in in front of something, it means not. So incomplete means not complete. If something is inadequate, it is not adequate. Okay? So incomplete dominance, neither allele dominates the other. Okay? So you can think of them as both being dominant. If we use the same analogy, A yellow, B blue, offspring with one of each are neither yellow nor blue. They're a blend. They are green. Okay, because yellow and blue make green. I hope you will agree. Okay, so genes that show incomplete dominance, if the alleles show incomplete dominance, they blend together to create a new trait, which is not seen in either of the parents. Okay? So a yellow parent and a blue parent make a green parent. It's a totally different trait. Here's an example of that. PTC, tasting. Now you may recall in class us testing whether or not we could taste PTC, and I use that as an example of a dominant versus a recessive allele, but actually it's not completely dominant. Okay, in fact, it is incompletely dominant. The gene is here, TAS2R38 is the code. It stands for taste2 receptor 38. This is the protein. Of course, you all recognize it, I'm sure, by sight. The alleles are either tasting or non-tasting. If you have two tasting alleles, you get a really strong taste. If you have two non-tasting alleles, you get no taste. But if you get one tasting and one non-tasting allele on your chromosomes, you taste it a little bit. You have partial taste. Okay? So incomplete dominance, the two alleles, neither can dominate the others, and so they blend together to create a new trait. In this case, it shows partial tasting. It's not as strong as the full tasting, and it's stronger than the no tasting. It's in between. It's a blend of both traits. So this is called incomplete dominance. The third form of inheritance is called co-dominance. If I add the prefix co in front of something, it means together. So if I am cooperating with someone else, I'm operating with that person together. So co-dominance means that the two alleles are dominant. Again, neither one covers up the other, but instead of blending, okay, they are expressed together at the same time. Pardon me. So if our analogy of A yellow and B blue, an offspring with both would neither be yellow nor blue, it would be both yellow and blue. Okay? Notice that the yellowness is the same as the yellow parent, and the blueness is the same as the blue parent. It's not a blend, okay, like we would see in incomplete dominance. It's the fact that both are shown together at the same time. That's called co-dominance. A lot of students in Science 10 and indeed in Life Science 11 struggle with these two because they don't really think about what the words mean, and as a result they forget which one is the blend and which one is the expressing together. You just have to think about it. It makes sense if you understand the words. Okay? A good example of this is feather color in some breeds of chicken, where you have a black allele and a white allele, and when you cross the two together, you don't get gray like you would if it were incomplete dominance. You get black and white feathers, and this is called erminette. All right, so here's an erminette chicken, right? The key thing here to remember with codominance is that if you look at these feathers, they are both black and white at the same time. They are not a blend of black and white, which is gray. They are black and white. That is the characteristic feature of codominance. Both traits are expressed together. They are co-dominant. Final form of inheritance is called sex-linked. If there is a gene found on one of the sex chromosomes, in humans those are called X and Y, if there's a gene found on one of those chromosomes, it will be inherited differently in males and in females. Okay? If that is the case, it is called sex-linked inheritance. So what happens here is that your biological gender, which chromosomes you inherit from your parents, determine whether or not you can inherit the alleles or not and get the traits. So the result is an unequal or skewed inheritance based on gender. So if we have a yellow father, X and Y is a father, and a blue mother, let's say that the offspring are two blue females, one blue male and one yellow male. Okay? This doesn't make sense according to any of the other forms of dominance unless we understand that the X or Y chromosome is carrying that gene. Okay? Now, just a quick aside here, sex-linked genes can be completely dominant, they can be incompletely dominant, and they can be codominant. Okay, so you can have a sex-linked, incompletely dominant gene. And you will see examples where, in the practice problems, of things that are more complex like that. But we usually look at simple ones. A good example of a simple sex-linked inheritance is a syndrome called Coffin-Lowry syndrome. It is caused by a dominant allele found on the X chromosome. Okay? And it results in a variety of mental and physical differences. Coffin-Lowry syndrome. So let's say that we have a father, a male, who has a dominant Coffin-Lowry allele on his X. He does not have anything on his Y, remember, because X and Y are not the same chromosome. And he has a partner who has two normal recessive alleles on her X chromosomes. Okay? What would happen if they had children? Well, if you do a Punnett square, you see this. The father passes on his X chromosome to the female offspring, and he passes on his Y to the males. Okay? So the result would be that both of the female, all the possible female offspring, I can't say both because remember it's always probability, all of the possible female offspring would have Coffin-Lowry syndrome because they would inherit a dominant allele from the father that has the syndrome and a recessive allele from their mother. Okay? So this is sex-linked because it appears only in the females and not in the males. Okay? If these two partners had 10 boys, none of them would have Coffin-Lowry syndrome. Okay? But if they had any daughters, they would. So that is an example of sex-linked inheritance. Okay. Let's do some examples. All right? So for incomplete dominance, I'm not going to do an example of complete dominance because you can see those in lesson 1.2. Go and have a look at some of those if you're interested. Incomplete dominance is the first one. PTC tasting is the one that we looked at. So here we have a woman who cannot taste PTC and a man who can slightly taste PTC. And the question is, what is the chance that their children will be unable to taste PTC? So when we were doing completely dominant examples, we represented our alleles with capital and lowercase letters. Typically with incomplete and codominance, the standard way of doing it is to represent the alleles with different letters, different capital letters, because they're both dominant. Okay? So we can choose whatever we want. Let's use T for tasting. Makes sense to me. And then let's use N for non-tasting. That also kind of makes sense to me. Okay, T and N. You can use whatever you want. You could use X and Y, but that would be confusing. So really that's the only hard part. Remember, is setting up the alleles of the parents. So we need ourselves a Punnett square. The parents from the question are a woman who cannot taste PTC. Okay, so that means that she has non-tasting, and she is crossed with a man who can slightly taste PTC, so he has a tasting allele and a non-tasting allele, like that. Okay, the order isn't important. You could put N first, but I just happen to put T first. So a woman who cannot taste PTC and a man who can slightly taste PTC. Those are their genes, their alleles, I should say. Okay, we put them into our Punnett square and then we answer the question. So starting with, I'm going to put the female on the top. It doesn't matter. Put the male on the side. Yeah, so now I'm going to fill in my square. These ends go in this column, going down, down. These ends go down, down here, and my T's go across, across. And this end goes across, across to N. Now I got my order wrong here, N and T, but again, it doesn't really matter. Now the question asks us, again, make sure you're answering the question. If you just write 50% slight tasting, you're not correct. That's not what the question's asking you. What is the chance that their children will be unable to taste PTC? So if we look at the alleles of these two offspring, they inherit one tasting allele and one non-tasting allele from their parents. Therefore, they can slightly taste. These are slight tasters. And then these two offspring inherit a non and a non, so they cannot taste. So the answer is that 2 out of 4, or 50%. So the chance that their children will be unable to taste PTC is 50%. Okay? So a couple of slight differences here in terms of how we represent the alleles and how we interpret the results, but the basics of how we do Punnett squares do not change. Okay? We still set up the square the same way. We still put the parents on the sides, fill in the rows and the columns according to where they are. We make sure our offspring inherit 2 alleles. Okay? All that stuff remains the same. Let's do a complete, or I should say, let's do a codominant example. So for codominance, we used black and white feathers, making erminettes. So let's use black, capital B, and let's use, it's a nice contrasting color with black, green. White. Okay, again, 2 capital letters because they're codominant. I'm color coding it so that it's really obvious where the alleles are. It's always important for me that I make things as obvious as possible. Look at all those straight lines. Wow. What a pro. So, what are the parents? It tells us here that we have a black chicken. Okay, that is going to be 2 black alleles, right? Because if it had a white, it would be different. And an erminette chicken. What's that erminette chicken going to have? It's going to have 1 black and 1 white. Like that. Okay. So we set up our Punnett square. It doesn't matter where they go. I'm going to put the black on this side this time, and the erminette's going to go on the top. I'm going to fill in all my things, so these B's go across, 1 here, 1 here. These B's go across, 1 here, 1 here. This B goes down, and this W goes down. Switch to my green. Like that. Okay, there we go. Now again, you might be tempted to say 50% black. That's the answer. Nope. This question asks us quite clearly what percentage of their children will have white feathers. Looking at our Punnett square, these 2 are black. Okay. And these 2 are erminettes. So the answer is the percentage with white feathers is 0%. Okay. Doesn't matter how many children they have, they will never have a white offspring. Okay, they can't. Final example. Here we have an example that is sex-linked, and we know that right away because we see that it's found on the X chromosome. Okay? Remember what I said when I was introducing sex linkage, sex-linked inheritance, that if it's found on X or Y, that makes it sex-linked. Okay? In this case, fortunately, it is a completely dominant example. Okay? So we have Coffin-Lowry being completely dominant over a recessive allele. All right? Here, our terminology needs to be a little bit different. And that's because in order to determine sex-linked inheritance, we need to keep track of two things. We need to keep track of whether or not the offspring get an X or a Y, okay, which determines their biological gender, male or female. And we need to keep track of which alleles they get on those X's and Y's. All right? So the easiest way that I've found to do that is to use X and Y. Okay. And then to represent with a superscript, a little letter that's above but smaller than the other letter, to represent the alleles that you're keeping track of. In this case, we have a dominant allele that's causing the disease. So let's use, well, I'd be tempted to use C, but C is the same in upper and lowercase. So yeah, let's use L. Okay, some students use N for normal or something like that. Let's use L. So in this case, we would have capital L, lowercase l, and nothing on the Y. Okay, so this is the Coffin-Lowry. This is normal, recessive, and this is nothing, okay, because it's the Y. All right? Set up our Punnett. Think carefully about what our parents are going to be. We can write out automatically XX and XY. You know what I'm going to do? I'm going to make a slight change. I'm going to use color here. I'm going to use red for Coffin-Lowry. And I'm going to use, let's be Christmassy. Let's use green. Okay? That'll hopefully make it easier to see the difference, even though the letters are different. So we know one of them is going to be female, one of them is going to be male, so we can do that right away. Then all we do is we map our alleles on. So the question tells us that the woman does not have Coffin-Lowry syndrome, so that means she must have two recessive normal alleles, otherwise she would be affected by this syndrome, wouldn't she? So that's her. And a man who does have Coffin-Lowry. Okay, so that means that he has the capital L. Okay, don't write anything on the Y because it's not on the Y, it's on the X. That's why it's sex-linked. Now all we got to do is put this on our Punnett square. Doesn't matter where they go. Let's put the female on the top and the male on the left-hand side. We need a red L there and two green L's here for the female. Okay? Same process inside here. This X goes down. Okay. being very efficient with my pen color changes here. I apologize. Let me just put them all in. And then I'll finish here. Okay. Okay, everyone see how that came from? These x's come down, these x's come down, that x goes across, and this y goes across. Okay? Check and make sure that you have 50% males, 50% females, because if that's not the case, then you know you've done something wrong. Okay? 2 females, 2 males. The question asks us, will any of their children be affected? Okay? These 2 females each get 1 recessive allele and 1 dominant allele, and that means that they do have Coffin-Lowry syndrome because it's dominant over that. The 2 males each get a normal allele from their mother, so they will not have Coffin-Lowry syndrome. Okay, so let's answer the question. Yes, 100% of the female children will be affected. Now, sometimes students will be confused by me saying 100%. Mr. Bullard, it's only 2 out of 4. Yes, but I'm not saying 100% of the offspring. I'm saying 100% of the female offspring. Oh, that's missing a word. There we go. If you wanted to say, yes, 50% of the offspring will be affected, that would be true as well, but this is a more precise answer. 100% of the female children is more precise than 50% of the children. Right. They're both correct, but this is a better answer. So this is why it's X-linked. It's found in the females, but it's not found in the males. Okay? Take my advice, use this notation with the X and the superscript letter, and I think you'll find that that's the best way to do these examples with sex-linked inheritance. Okie doke. Let's wrap up here quick. I just want you to think about Punnett squares in the bigger picture, briefly. And so, some simple math. If a Punnett square for 1 gene is 4 cells, that's 2 by 2, how big would a Punnett square for 2 genes be? In order to understand this, you have to understand a little bit about probability, which most of you don't, I know, and also a little bit about exponents. Okay, this is actually 2 to the power of 1 and 2 to the power of 1, which is the same as 2 times 2. Okay? A Punnett square for 2 genes is 4 by 4, or 2 to the power of 2 times 2 to the power of 2. Okay? And that makes a total of 16 possible allele combinations. So the number here, the superscript, the exponent, is the number of genes. Okay, so for 1 gene it's 1, for 2 genes it's 2. How big would a Punnett square for 3 genes be? Well, it would be 2 to the power of 3 times 2 to the power of 3, which is 8 times 8, which is 64. Okay, and so it goes 128, 256, and so on. How big would a Punnett square for a human being be? Remember that human beings have approximately 27,000 genes. Okay? So in other words, what is the Punnett square for your parents, for your family? Okay, of which you are 1 square. The answer is 2 to the power of 27,000 times 2 to the power of 27,000. Okay, that's how big the Punnett square is for your parents. And you represent 1 of those. Okay? This number is really, really big. Just to give you an idea of how big it is, this is 2 to the power of 27,000. Okay? I don't even want to start reading off this number. It is so, it's beyond astronomical. In fact, there are no things in the universe that are this big. okay? And that's just one of them. You then times it by itself, okay? That's the probability of you existing, okay? So what is the chance that your parents would have two identical children? If they kept having children, would they ever have you? Well, of course they couldn't. Students will always say then, Mr. Bullard, what about twins? Well, if you know anything about twins, you know that they are identical, okay? They're a single egg, a single fertilized egg that divides, okay? Non-identical twins occupy different squares on this enormous Punnett square. What I'm trying to say is that you are unique. There can never, ever, ever be, no matter how many humans ever exist in the future of humans, those alleles will never come together in that combination again. Okay? You are unique. There can never be another you. Now what I'd like you to do, please, is to fill in your notes for 1.3 if you haven't done so yet. And then if you feel confident with Punnett squares, if you've practiced 1.2 and you feel like you know what you're doing, I recommend you try the practice questions that follow lesson 1.3. Remember the notation for incomplete and codominance using capital letters for the alleles, and the notation that I showed you for sex-linked inheritance, where you have the X or Y, and then you use the superscript on the X or the Y for the allele. Okay? Lastly, know that we are working towards our next quiz, and that will cover lessons 1.2 and 1.3, learning targets 1.2 and 1.3 as well. Make sure that you are practicing this and that you understand how to do Punnett squares for all of the different forms of inheritance, and you will be ready for the quiz. Thank you for your attention. Bye for now.